am Livia. I work for the University of Rome Tor Vergata together with Fondazione Human Technopol in Milan. And I am the coordinator of a database called Senor, which will be part of the topic that I'm going to discuss today with you. So the title of my session is Methods in Building and Analyzing Biological Networks. So this will be the topic of this webinar. And if you need to contact me during the course or after the course, this is my email address, so livia.perfetto@fht.org. So let's start. So today, this is a very dense course, so we are going to go across a number of topics, so ranging from different types of biological networks. So we're going to introduce the main topic of the day. And I also will describe very basic graph theory concept that I believe can be useful for you, for your research in the future. I will also introduce you to way to build biological networks using public repositories. I'm going to showcase IMEX and Senor databases. Finally, I will also describe very briefly Cytoscape, which is one of the most popular tools for network analysis and visualization. So I need to apologize in advance because this is a very dense course. It usually takes between 4, 5, and 8 hours to go across all these concepts more accurately. But because of a matter of time, I'm trying to be, I might need to rush. So if anything is not clear, just type the question and I can try to clarify the concept again. So let's start with the different types of biological networks. So in biology, we have many different type of biological networks. So networks are a concept, are a way to represent interaction. So we might have, in this sense, a lot of different type of interaction, because we can also have interaction between animals or human beings, and like in ecological networks. We can also have interaction between cells or tissue, like in neurological networks, but we can also have interaction among molecules. So basically protein or metabolites or genes and so on and so forth. So this group here in red has something that we call molecular interaction networks, where the entities involved in the interaction are molecular entities, and will be so we will deal mainly with this type of interactions. So before we start, let me introduce you with some very quick definitions that might be useful for the webinar. So let's start with the definition of protein-protein interaction. So protein-protein interaction represent the physical and selective contact occurring between a pair of proteins. So it might happen in certain region, in a defined context, and they tend to be specific. Usually, protein interaction involve the transmission of the information from a regulator protein to its target. Another important concept for this presentation is the concept of interactome. So we defined as an interactome the totality of molecular interaction occurring in a given context. Like the mitochondrial interactome indicates the ensemble of potential or known characterized protein-protein interaction occurring in mitochondria. An important thing for this presentation is the type of mathematical abstraction that we use to represent network, which is called graph. So anything that represents an interaction in this presentation, so everything that we call network, can be described in a mathematical formalisms using graph abstraction. So we tend to confuse these two concepts, but in reality, a network is one of the many ways to represent interactions, and the graph, which is this formalism, is one of this particular way. In a graph, every interacting partner is indicated with these dots, like here or here, which are technically called nodes, and interactions are indicated with these lines connecting nodes that we usually call edges. So why networks are so important? Basically because we can obtain from network additional information that we wouldn't get by analyzing the individual components. So it's something that we call an emergent properties. So when we analyze all the components together, we can obtain additional information. So for instance, just by looking at this network here of interaction, I can already by eye appreciate that there are more important proteins in this network, like this orange protein here, because they are involved in many connections. These proteins here, which have a high degree, are usually called hubs. Also, I can find in a network community of entities, of proteins that are more connected to each other compared to the rest of the networks. And this protein can, so these communities can refer to protein belonging to the same pathways or to protein complexes. But networks are also useful to identify new disease genes or to simulate, in silico simulate the cell behavior, or to formulate new hypotheses about a protein function. Let me give you an example. This is something that we call guild by association. So let's imagine that we have this red protein here. So this is a guy. We know nothing about it. So if we look at the individual red entity, we cannot really estimate the function or predict the function of this protein here. But when we look at its neighborhood, so the proteins to which it interacts, and we start analyzing the functional role of these guys as well, we can start hypothesizing that this red protein here might also have something to do with transcriptional and cell cycle control. Obviously, this is just an estimation, so we cannot be sure. We need to confirm this hypothesis, but at least we can narrow down the search space for the cellular function of this entity. Another important reason why we want to use network is that there is a huge number of graph theory formula and concepts that are applied not only to biological networks, where interactions are between biological entities, but they also benefit from a wide range of theories that are applied to social networks as well. So where basically we represent interaction between people. So this is a very studied discipline. There is a new discipline in biology, in systems biology, called network medicine or network biology, has proven to be very useful and very powerful in analyzing the biological behavior of a system. Let me now introduce you some basic theory, graph theory concepts. So when we say graph theory, we mean a number of properties that networks have either on a local or a global rules. So when we look at a network, we can identify some properties that belongs to the local nodes, to the local entities. So the tiny dots here, like in this case, the degree, we can estimate the degree of a node by just looking at it. Or like the shortest path, which is the minimum number of steps required to connect two proteins. But we also have more global properties of network. So properties that we can resume by analyzing the entire network. Like some of them, we are going to see more in detail some of them. Oops. So let's start with one of these properties. So one of the global property is that the biological networks, and in particular protein-protein interaction networks, are scale-free networks. So what does it mean? Well, basically it means that the number of connections is not equally distributed over the nodes of the network. In other words, it means that we have few nodes called hubs that have lots of connection, and a large number of nodes which have a small number of connections. So the distribution of interaction is not equal, is not random. This is very important because these hubs have been proven to be relevant proteins, central proteins, important proteins. Like for instance, usually many cancer-linked proteins like TP53, MAP kinase 1 are hubs. This means that they have a greater impact when removed from the network. So let's imagine that we have a mutation that impacts one of these proteins. So we need to remove them from this network. We would have a stronger impact compared to the removal of a more peripheric protein. Another important global property of a network is the centrality or transitivity. Basically, this can be applied. So this graph theory concept can be applied either to proteins or to edges, so interactions, and indicates how often a shortest path passes across that edge or that protein. So how relevant a given node or a given edge is to connect different areas of the network. This is also another way to estimate the importance of a protein in the network. And we can estimate that proteins, nodes that have a high centrality, if removed from the network, would cause major disruption in the way the different areas of the network can communicate with each other. Another important global property are the communities. Communities are densely connected areas of the network. There are a number of algorithms developed to identify community. Community are important because they have proven to be associated with specific biological processes or pathways, or with well-characterized complexes in a network. For instance, the components of the proteasome or of the nuclear pore complex tend to cluster to form communities in a protein-protein interaction network. So if we have a new protein that we don't know what is its function, but we know that it falls in the cluster of a given complex, we can start hypothesizing that this protein belongs to the complex as well. So we have now described some major general concepts. Let's go a bit more toward the practical bits. So how can we build biological networks? So I hope I have convinced you that biological networks are very useful, powerful, and interesting. So now let's see how we can build a biological network. They exist nowadays a huge number, tens of thousands of biological databases, and they annotate many different things. So I cannot describe all the molecular interaction databases, and I have picked two databases, Senor and IMEX, as example of protein interaction databases. However, we can, without going across all the public repositories available, I can describe few of the most popular databases that are used to annotate signal transduction. So molecular interaction in general. So I call signal transduction, but I mean molecular interaction. So the plethora of interaction type occurring between biological entities. I am particularly interested in signal transduction because this is the main topic of my research, where I'm very much interested in understanding and get new insight about the way a cell is able to respond to its internal or external environment by activating the cascade of the signaling. So, going back to our public repositories, all the repositories that annotate molecular interaction can be categorized in these 4 different groups. The different groups differ by the way, the formalism that each type of database adopts to represent the same biological information. So let's imagine that we want to represent the regulation of ELK1 by these two proteins, UBC9 and ERK, and the way ELK1 itself activates CFOS. The molecular interaction databases adopt 4 different formalisms to describe this information. We might have protein interaction network, where basically this information is represented by the plane, the symmetrical interaction between all the components of this network. So we don't have any directionality, any mechanism, so it's just plain interaction. Display interaction. We also have activity flows that are similar in topology, so they are similar in topology to protein interactions, but in addition to that, they have directionality, so we know which protein is the regulator and which one is the target, and they also have the effect, so like this T-shaped edge here indicates an inhibition, while the arrow an activation. These 2 types of interaction model can be described by graphs because they meet the requisites of a graph. So we have nodes connected by edges. We also can also have 2 additional types of representation. So the process description, where actually every node can be represented in multiple ways. And we might have regulator that acts on the transition of the node between different states. So these are not proper graphs, so we are not going to analyze in more detailed process description, nor enzyme substrate interaction, because they cannot be represented by graph. But I want just you to know that they exist. They are very popular databases like Reactome, KEGG for the metabolism, or Navicell, or Phosphosite Plus for enzyme substrate. For Phosphosite Plus is great if you want to study phosphorylation, like phospho-ELM as well. So going back to our graphs, so I'm going to just discuss a bit more molecular interaction, so interaction network, and causal interaction. So the most popular databases that annotates interaction, like this symmetrical interaction, are Intact, Mint, DIP, Menta, and String. So String is the largest one. They are very useful because they have the best coverage. So they can have very high coverage in terms of interaction or proteins of species. They also have a lot of metadata and the benefit of decades, because they're more ancient, of network analysis tool. The cons of using interaction network is that they don't provide any information about the directionality and the mechanisms, the sequentiality of the interaction. So they are a static picture of interaction in a cell. And they also lack information about the way the signal is propagated toward the cell. So one of the most popular databases that annotates protein-protein interaction is Intact. Just to give you a hint, it has more than 1 million of interaction annotated, and it also annotated at a very high standard called the IMEX standard. Intact has the advantage of gathering together a number of sister databases like Mint, UniProt, which share a common scope and a common aim to annotate molecular interaction. Then here we have activity flow or causal interaction or logic interaction. They are all synonyms. Again, here we have a graph representation of biological events where we also have the directionality and the sign. So these activity flow network, which are very similar in structure to protein interaction network, are very suitable for modeling approaches, so they can be turned into Boolean models. They have more information because they also they provide information about how the flux of information is propagated toward the cell. And they also can benefit from graph theory principle. The problem here with activity flow databases is that they have limited coverage. So they are way smaller than the protein-protein interaction counterpart. So if Intact has 1 million of interaction, the 3 databases together don't reach 100,000 interactions. So they have very limited coverage. So there are many ways to use causal interaction. Just to give you a very quick hint, they can be used by any type of biologists, clinicians for the interpretation of omics data, just from mere consultation, for simulation studies, and so on and so forth. So before we move toward the more practical bit, I just want to spend a few words about Senor. Senor is a database that annotates causal, logic, activity flow relationship between biological entities, mainly proteins, but we also have complexes, phenotypes, stimuli, and so on and so forth. So the model adopted is the model of activity flow. So every protein is a binary. Every interaction is is composed by a binary pair of proteins connected by edges. The edge indicates either the directionality and the sign, so the upregulation or the downregulation of the target. Senor is a manually curated database. It means that every information contained in Senor is extracted from a piece of evidence from the literature and is also supported by a short sentence that supports the interaction. Every interaction in Senor is merged together to form a large interactome. So Senor contains 31,000 interactions. Everything is also curated to the human data, so it's human-centric. So you don't have other model organism, the curation done for other model organisms. So this is the web page. So here is the new website in its beta version. So as you can see from the web page in Senor, we also have a number of related projects. So we have also we are also involved in the curation or more specific interaction dealing with COVID-19, with cancer, muscular regeneration. As mentioned before, Senor is human-centric, meaning that everything is curated to the human proteins. However, we also computationally provide a mirror of the human interactome for the mouse and rat proteins. So just to give you an example, we can search in Senor. We can perform many different types of searches. We can search for individual proteins, like we can search for the protein Notch2 and Notch3. We can decide to search for all the interaction involved in Notch2 and Notch3. We can search for direct connection between the two proteins. We can search for shortest path connecting Notch2 or to Notch3, or we can decide to search direct connection plus bridge proteins. So let's try with this case. The results of this network are all is visualized as a graph, as you can see, where you have the two proteins, Notch2 and Notch3, and all the bridge proteins. So all the proteins that bridge Notch2 to Notch3 and vice versa. Every interaction in Senor is displayed in this form of a graph, where the graph is distributed over 4 major cellular compartments. So we have the extracellular space on the top, the plasma membrane here in yellow, the cytoplasm, and the nucleus. So you can, this is an interactive viewer, so you can decide to show the score, to hide the score. you can decide to show just the up regulation or different type of interactions. Senor also annotates pathway, canonical pathways captured from the literature. So you can go to the pathway browser and select and visualize one of these pathways. So let's see, for example, this is the autophagy. Also here, you can see the graph viewer with all the interaction displayed. As mentioned, we have different type of entities. So most of the interactions involve proteins, but we also have stimuli, phenotype, complexes, and other another entity type not displayed here, like small molecules or protein family or chemical compounds. Going back to the presentation, now I want to show you, I want to introduce you and jump to another topic, which has to do with Cytoscape. So why Cytoscape? Because Cytoscape is the most popular tool to analyze, visualize, integrate networks with experimental data. So Cytoscape is open source, so I forget to update here, but we are now at the 3.9 version of Cytoscape. So its content is constantly updated and ameliorated and improved to have better visualization and better analysis. Why Cytoscape? Well, because it can make use of several visual features that can effectively highlight key aspects of a network. So networks are very useful because they very intuitively teach you about the importance of a protein. So it's just by eye that you can see community, you can see how the information is flux from a protein to another. So they are very intuitive, which is very useful for us. In addition to that, Cytoscape also permits to integrate global data sets and functional annotations. So basically, it allows you to integrate in your network also additional data like gene expression or, I don't know, additional features. Also, in addition, it also can be considered as a framework where additional analysis and modeling tools are available and integrated. Especially, in particular, there is a number of applications of apps that can be developed by anyone that are integrated in in Cytoscape. So they can be used and imported in Cytoscape to have more powerful analysis. There is a large number of variety and a type of plugins and apps. They are very flexible, they can adapt, and they are all integrated in Cytoscape. So it's very prêt-à-porter. And in addition to that, so you can use Cytoscape to search directly for public repositories. So either Intact and Senor have their own Intact Cytoscape app. So you can query the databases directly in Cytoscape, where you can then use all the visualization features in Cytoscape to modify the networks and import your attributes. A network, how can you build a network in Cytoscape? Well, basically, you can just import your table file from your laptop, so from your desktop, but you can also query public databases integrated in Cytoscape, like you can use the Scikit web client. You can use Index, which is a collection of networks. And you can use ad hoc apps like the Intact and the Senor ones. So we are going to briefly show you how to use these two apps. Before I start, in the future, if you need to test these apps, you will need to first install Cytoscape. You can freely download it from this URL. And you can then install either the Intact app or the Senor app, just by going to following this instruction here. Once you have these apps installed, you should be able to see them by just seeing these for intact, these two logos here, and for Senor, these 4 colored logos here. So before we start, I just want to show you a little bit, for those of you who are not familiar with Cytoscape, how is its interface. So Cytoscape is very popular because it's very intuitive and very flexible, and it's very nice because you can easily visualize a network. So the first more impactful panel in Cytoscape is the visualization panel, where you can effectively visualize the network. This is just a visualization. On the left, you have a summary of the number of nodes and edges displayed in the network. It's important to stress that the network, the graph that you see, is just a representation, one of the many representations, as I mentioned at the very beginning of this webinar. Part of the network displayed is contained in the tables down here, which namely the nodes table and the edge table. In particular, the edge table is the true real network that you can use. In addition to nodes and edges in the edge and the node table, you also have a number of attributes that can help you to filter and modify the layout of the network displayed. In addition to that, you also have a number of additional panels. Here in the middle, you have a number of shortcuts that allows you to upload a network, zoom in, zoom out, refresh the layout. On the very top, you have a number of tools that allow you to modify the layout of the network, to integrate additional apps from the App Store, or to perform network analysis. While on the left here in the panel, you can have the tool to filter the network, to manually modify the style. Last but not least, applications that allow you to search for a network are integrated in this search box here, so that you can directly do your query to Intact or Senor from the Cytoscape interface. So, for example, if I want to search Intact for interaction involving proteins, I can go to I can select this panel here and select the Intact fuzzy query, type notch 3, and click on the search icons. So basically, Intact has a way to solve term ambiguity, just in case you're, in your case, like in this case, notch 3 matches more than one protein. Once you have detected the protein of your interest, you can build the network. The network visualizes a graph. In this case, in Intact, different colors indicates different species, just because Intact is manually annotated from the literature. So sometimes experimentalists have mixed evidence of, a clone of TP53 interacting with a rat MDM2. So you can use this network, you can manipulate and, play with the network. Just to give you a more practical example of how you can use Senor. So you can use Senor to perform different queries. So if you, once you have downloaded the app, and you can download the app by using the stop bar over here. Once you have downloaded the app, you should see these 4 logos here. And as you do with a web resource, you can perform exactly the same type of searches here in the app. So you can search for notch 3 and notch 2. Also, in this case, we are going to select the bridge proteins. You can decide to pick human or mouse or rat interaction. And once you have, you can click and you can display exactly the same network that you had before. An interesting thing is that you also have all the information annotated in the Senor resource, in the node and in the edge tables. Another way, another type of search, we can again search for the for a pathway. So we can decide to search for the autophagy. So the cool thing is that you can here modify the layout. So now I cannot see my top bar, but oh, how annoying. Yeah, I'm sorry, I cannot see my top bar because of the, well, nevertheless. So you should be able to do, yes, network analysis. You can do analysis for the network by click on analyze the network. Like in this case, it's asking me to perform basic graph theory analysis, or searching for the degree of the nodes, for the diameter of the network, and the distribution of the interactions, and so on and so forth. In this particular case, my graph is a directed graph because they have directionality. And I can use all the information, so all the analysis have now been uploaded here in the bottom part as attributes of the node and the network. So the betweenness centrality, which I mentioned to you before, is now displayed as an attribute of the node here, thanks to the network analyzer tool. I can then decide to modify the style of my network by taking into consideration the network centrality. Like, for instance, I can decide to modify the size of my nodes by as function of the centrality. So I see, because these are numbers, they can be interpreted as a continuous value. So, and I can play with it, you see, by to show what are the more relevant nodes in my network. I can also use different types of layout. Like, I can pick the hierarchic layout to show really how the flux of information is propagated. And now you can see how intuitive it is to see that this protein here forms really the major stream of the pathway. Okay, let's go back to the presentation. So, as mentioned, we can use Senor to perform different type of searches. So I went very quickly across the different type of searches, but basically the all interaction allow you to search for all the interaction involved in the two proteins. The connect search allow you to search for direct connections between the two proteins, while we already saw the include bridge entities type of search. We can also search for pathways. We can play with the visualization tool and with the style. And another thing I want to show you is example. So it is to suggest you how a flexible Cytoscape is. So you can really easily and very quickly build a network querying public repositories, linking proteins of your interest. You can use additional apps like the EMCode app to search for clusters and communities, or the path linker to search for shortest path, or the Clugo tool to perform functional enrichment. So everything is very much integrated into Cytoscape, and you don't need to jump from a tool to another. So you can do anything within Cytoscape. If you, as mentioned before, this was a very dense presentation, and I apologize for all the concept, so which I rushed, and I am already sure that perhaps something was unclear. So if you need additional information, you can find the documentation for Senor in the supporting material in our publication, but also if you need a tutorial for the app, you can find tutorial either for the Senor app and documentation, either for the Senor app and for the Intact app. So I think that with that, I am more or less done. So perhaps we can move to the question.
All right, so so the first question is about, okay, so I'm wondering if lack of interaction is because it has not been studied or it has been proven that those entities really do not interact.
That's a very good question, and is a long-standing issue for public repository that annotate molecular interaction. There might be tons of reasons why an interaction is not there. So as you mentioned, it can be that it has been demonstrated that the proteins do not interact, but it can also be that the protein, so the interaction has not been yet demonstrated, or perhaps not yet been annotated in public repositories. So it can be many different reasons. There have been attempts to annotate the negatome, so the interaction proved to not occurring. But to be honest, it has remained an orphan project, and it has not really been fully developed for lack of resources. So thanks for this question.
All right. So the next question is, can we use list of proteins in Senor?
Yes, of course, as mentioned. So I gave you the example of the notch 2, notch 3, but it can be a list of proteins, so many. I think there is a limit of perhaps for 500 proteins. But if you need to search for that many, what I suggest you to do is, instead of performing the search using tons of proteins, you can download the interactome by clicking here, and then filter the interactome for the protein of your interest. So if there are too many, like 2000, I would suggest to go for the interactome. But if you want to search for a reasonable list of proteins, you can also use the entity query and connect.
All right. So next question is, how can we know whether protein is upregulated or downregulated unless some values are given?
Well, if you mean upregulated or downregulated in general, like by looking at this expression in a biological context, the answer is that we cannot not know by just looking at Senor. So we need to look at that protein in the context of its interaction to have this information. So Senor does not capture expression of proteins per se. It captures molecular regulation.
All right. So the next question is regarding the guilt by association.
Yes.
Let's say node A has a degree of 10. Each node has 4 types of class labels. How do we assign the labels to node A? Is it based on their degree, or should we be assigning the label value which at least 50% of the degree has? So
well,
how to approach this problem?
I am not 100% sure I got the question, but if I got it correctly, so of course, this is not so easy to assign a molecular. Well, you don't really assign molecular function. You narrow down the possibility of molecular functions that you can assign to that protein by using the guilt by association concept. But of course, what you really need to do is to apply a statistical test that makes your inference statistically significant. So it cannot just be done by looking at these interactors. So it was a simplified way to see the potential of this method, but really you need to apply some test.
Okay, so the next question is, can I input a matrix of gene expression correlation between genes derived from RNA-seq data into Cytoscape to run the network analysis that you are showing?
Yes. Yes. I think that you can import metrics, but I have, if I'm not wrong, to import metrics in Cytoscape, you need to pass across the R package, an R package. So you need to pass with the iGraph tool. iGraph tool has a function called something like RYC3. I need to search to give you further detail. But with this function, you can import the metrics in a graph and then the iGraph object into Cytoscape by using this function. But in the end, you can have a Cytoscape layout from a matrix. So the answer is yes, it's possible.
All right, as next question, the way string and intact provide the ability to download the human protein interactome, does Senor also provide with that feature?
You can download the full interactome, yes. Yes, you can either download it in Cytoscape here by the Senor download interactome, but you can also download it from the web resource by going to the download page. So if you go to the download page, you can select the entire interactome from either human, musculus, or Rattus norvegicus. Does it reply to the question?
I think so. All right. This question is about Cytoscape. How can we compare two networks in Cytoscape? Is it possible to do that?
Yes. So what you can do is, I'm sorry, because I have this bar. Okay, now I can move it. So sorry. So you can have more than a network. Like, for example, I can have. Let me give you an example. Another pathway. So let's speak, for example, the MPK pathway. So with these tools over here, you see on the top bar, you can compare network. So you can compare, the tool is called merge network, but in reality, you can also check for the union, for the intersection and the difference between two networks. So in this case, for example, I want to check for the intersection between MPK and the autophagy pathway, and I do that. So in this way, I'm sorry for the style. This is a little bug that we have. It loses the style. So in this way, we can see what are the molecular interactions that are in common and share between the two networks. The tool merge allows you also to analyze just the nodes. So if you go to the advanced option, you can also decide to compare just the node or the edges in the network. So yes, that's the way.
All right. Next question is about iGraph. I think you mentioned it in one of your answers recently. So what's your take on using iGraph R package instead of Cytoscape? Are there any advantages or disadvantages?
Well, iGraph is definitely more powerful in terms of computational application that you can see, and is more integrated within with other R packages. But to my experience, everyone that use a graph at some point needs Cytoscape to plot the graph, because the graph plotting is not the best. So usually the networks in the visual, from the visual point of view, are a little bit ugly and not really easy to manipulate. So with Cytoscape, they are cooler. Also within Cytoscape, you have all the apps. So it's very user-friendly, especially for people who don't have a computational background with programmatic skills. But if you have programmatic, usually I tend to use a graph when I do bioinformatics analysis, because so everything that you do in Cytoscape has to be done manually, even though you can use this command line here to perform more complex type of queries, but I'm not familiar with this command line here, so I cannot give you any hint on that.
Right, thank you. Next question is about metabolomics. So someone here writes, I'm interested in metabolomics. What are the possibilities and limits of Cytoscape for metabolomics?
Well, that's a tricky question. I'm not sure I'm able to answer because I'm just very recently starting working myself with metabolic data. I don't know, I cannot answer in general. What I can tell you is that now in Senor, we also have the metabolic data. So we have started the annotation of a number of metabolic pathway, like in this case, the catecholamine metabolism. So you have the all these metabolic, these small molecules integrated in the causal interaction network, forming causal interaction. So you have the enzyme, the precursor, the resulting molecule. So it's a new annotation. So the coverage is still very limited. In general, what I can tell you is that the problem with the metabolic data is that they are not very well integrated with signal transduction data. So to the best of my knowledge, there are just a few resources apart from Senor, like Reacton and KEGG, that annotates either signaling data and metabolic data. So it really depends on the analysis you want to do in reality. So if you want to have an integration of this information with signal transduction, so I think that the main limitation is the little coverage. So perhaps applying data-driven approaches rather than prior knowledge approaches would do better in your case.
Okay, next question is if there is a tool that also incorporates microRNA.
There are a number of resources that incorporate microRNAs. So Intact and Senor themselves, they both annotate RNAs. In particular, there is a big initiative, well, there is an initiative in Intact to capture microRNA target mRNA with a causal impact. But there are very large databases like MIR databases and which capture this information. In this case, I think that the best option would be to use another integrated resource called Omnipath. I didn't mention before because of with the time, very limited time we had today, but with the Omnipath, you can also select microRNA mRNA interactions. So in this case, you can select all the microRNA mRNA interaction extracted from the different resources. As you can see, you have Senor between the others, but you can decide to select them all and then display all the interaction already within Cytoscape.
All right, the next question is, I think, about databases changing with time. Sometimes using one version of database, we can find the interaction between two proteins, but after an upgrade to a new version, the interactions are no longer possible. Is it due to more evidence supporting that curation than previously does not hold true any longer? Does this link the evidence of PPI with experimental sensitivity and specificity?
So yes, that's true. So, well, yes. Manually annotation is an error-prone process because there are human beings behind which do their best, but obviously they're not, they can do mistakes. So when we spot a mistake, we tend to remove the mistake. So interaction can magically disappear from the database, but usually there is a reason behind that. Another explanation to this problem can be that sometimes large database like Intact also host direct submission, so pre-publication submission. It might be that during the reviewing process, authors refresh the list of potential interaction and update the interactions. So once these interactions are updated, some of them might disappear because they no longer pass the confidence threshold. So that's it. Yeah, I'm sorry. I apologize for this inconvenience. It's just an attempt to provide the best product at the end.
Okay, next question is about Senor. So how are the edge scores derived in Senor?
Well, that's a complicated question. So basically, we to score interactions, we apply two different methods. So everything is available here in this documentation, from the web page to documentation and the Senor score. So basically, for protein complexes and protein family interaction, we use a principal component regression model. So we basically feed this model with a number of scores extracted from co-expression, from other resources. And then we try to use this model to predict how, if two proteins belong to the same pathway. So we use pathway data as our gold standard to fit the model. And then we predict all the interactions in Senor. So anything that does not fall in these categories of interaction is assigned a default score. But as mentioned before, it's a bit complicated. So anything is available here in this document, in the documentation.
Right, thank you. Next question is about which database is more accurate and useful for protein-protein interactions?
Well,
if you have recommendation.
Yeah, I have recommendation, but obviously I am biased by my experience. So I want to stress the fact that there is no best database. There are databases that fit the purpose that you have in your research. That said, all the interaction database mentioned here, so everything that belongs to this IMEX, consortium here, so namely Intact, but also MINT, DIP, they all together form a consortium of very high quality interaction, meaning that everything is manually captured from the literature and everything is curated according to standard, so that we all the details of the interactions are captured, like the tags used in an experimental data setup. Everything is double checked by second curators, and everything undergoes an additional revision process that applies quality rules, so computationally. So we have two levels of quality check. So I would recommend that if you're looking for high quality information.
Thanks, Livia. And just to add that I'll be sharing these slides also later along with the recording. So if you can't find information right now or can't copy from the screen, you can check it later. All right. So the next question is, again, it's similar about database. Whether you know which database would be most useful for immunology research, which database to include for immunological protein interactions?
Well, immunological proteins are a bit tricky. So it depends on what type of immunological protein do you mean. So if there are like pathways, protein dealing with the immune response, it really depends on the type of data you need. Like in Senor, we have a number of immunological pathway. And I'm sure that in Intact as well, there is a large number of interaction involving like TNF response or something like that. Anything concerning antibodies or like major histocompatibility complex or anything that has sequences that changes frequently in a protein because of the highly variable sequences is a bit tricky to capture, because anything that we capture needs to refer to UniProt. And these proteins are not well captured in UniProt because of, well, it's the policy behind UniProt. So perhaps, so if you are interested in antibodies, specific antibodies, or these very variable proteins, it might be tricky. But if you are interested in the pathways, so the immune response pathways and stuff like this, any database like KEGG, Senor, Reactome, they have a very nice coverage of this information. Like here we have the inflammasome activation, the interleukin-1 signaling. Also we have the macrophage polarization, B-cell activation, antigen presentation. So we have quite a lot of stuff.
Thanks, Livia. So we are already over an hour, but if you don't mind, we can take 2 more questions.
It's okay. Couple of more.
Yeah. So, so this question is if there is any interaction analysis for bacteria or viruses specifically.
Well, okay. We I don't know if the question was meant for Senor. Since Senor, we have the COVID-19 project where we annotate also COVID related data, but this is a very limited project. On the contrary, Intact and the Intact database is specifically thanks to the effort of the MINT group. They have a lot of data on the bacteria interaction, bacteria, so host pathogens interaction for either for viruses, because they actually developed the tool called Virus Mint or Virus Menta, and for also bacteria host interaction. And also, of course, host bacteria and viral interactions. So if you're looking to this type of data, I would go for the Intact and IMAX resources. And you can access MINT data as well from the Intact webpage. So they are all well integrated.
Great. Okay, so this is going to be the last question. If there is no path between 2 genes at Senor or pathway, can we add it? And if so, how?
Yes, any submission is more than welcome. Any suggestion, any, please add this interaction. So you can either use the contact and send, you can either use the contact here, or, well, the report back perhaps is not the best, but you should be able to send me, drop me a line, by using this, this is my email address. The best things to do it would be by providing the minimum information required would be the interacting partners. So the best if they are the uniproteids of the proteins interacting, and an evidence supporting the interaction. Also, because we annotate causal interaction, it would be very nice if you could tell us if it's an up, so who's the regulator and who is the target, and if it's an up or a down regulation. But any submission is more than welcome, really.